Okay, great. Good afternoon, everyone. So I hope you are safe and well in these daring times. So what I'm going to talk about today is what we do with a computer simulation to address various complex questions. And in terms of complexity, I don't think your questions are less challenging. So it's comparable things, and I hope it will be useful. So I'll be talking about multiscale modeling of bionano interface. And first I mentioned the funding. So this project was supported in various aspects by several European programs. Horizon 2020, Smart Nanotox, most of all, where most of the research was done, where we looked at development of smart tools for predicting toxicity of nanomaterials. Then NanoCommons, we were developing infrastructure for managing information about nanomaterials and predictions. NanoSolvit, it's a nanoinformatics project where we apply ideas from bio and cheminformatics to nanomaterials, and biointerface modeling of bionano interface, where we're interested primarily in medical applications like drug delivery and diagnostics and medical devices. The team is presented here. So right now, five people there. David Power just graduated in August, got his PhD. Then Parinas and Kostas. Parinas is a PhD student, and Kostas is a postdoc working in NanoCommons projects. Ian worked in Smart Nanotox and now in NanoSolvit. And then Julia, she's working in biointerface projects, and Shada working on her own, but in collaboration with the biointerface projects. So they contributed different parts to this research. So my starting motivation is as follows. So if I try to define why bionano interface is important, I can mention several situations. I can talk about processing of biological fluids like milk or blood, where we certainly have a lot of interactions between the biological sample, biological entities, and the devices themselves. And there we have adsorption, binding of various molecules and cells to surfaces. And certainly the interesting part is coming up and in the nanoscale layer where they come in contact. When we talk about biomedical applications and bioengineering, about implants, prosthetic surfaces, and various other things that inserted and get in contact with live tissues, there we also have that. So we are asking how compatible those materials are, biocompatibility, or how the biofilm can grow on that, and how they can be integrated and incorporated into live tissue. And again, the important things are happening in the nanoscale layer at the interface. And when we talk about nanomedicine, the drug delivery, certainly a lot of experiences acquired here, where people manage to design particles for targeted delivery into certain types of cells. And then, of course, you have to engineer the surface. Again, the interaction at the bionano interface is important. So you need to know how it sits on the surface, how the grafted or adsorbed molecules influence the uptake and transport, and so on and so on. But in addition to that, toxicity was the driving force between the research I'll be talking about. Smart Nanotox projects was dealing mostly with the toxicity. Why are we interested at all in this? Because we have a lot of unintentional contact with the dust, which can be nanoscale size. And at industry, people are challenged with that since ages. For example, when you burn coal or steel plants, or it could be just stone grinding, silica construction materials, and so on. The dust gets into the lungs and can damage them. Nanomaterial production nowadays, carbon nanotubes are produced in large amounts, and various nanoscale components of advanced materials, nano-enabled materials like nanocellulose or titanium dioxide, zinc oxide, metal particle like silver is incorporated into various materials like textiles. And all of them can get into our body and lead to unexpected outcomes. And sometimes the outcomes are adverse. But also car exhaust, paints, construction material, cosmetics, all contain nanoscale components. So those contacts are unintentional, but in nanomedicine, we want to use the special ways of systemic transport of these nanoscale particles for targeted drug delivery, diagnostics, cancer treatment specifically. So many questions are related to this bionano interactions. And quite obviously, this when nanoparticle of a foreign origin, engineered nanoparticle, comes in contact with the live tissue, cell, or biomolecules, this is where interesting things are happening. So for nanotoxicity, a lot is known, and I'm citing publication which is 7 years old and citing just adverse effects coming from the interaction of nanoparticle with the cell. And it can be pretty harmful. Okay, then if we say that should we look indeed at the particle itself and interaction of inorganic piece of material with the cell? And in most cases, the answer is no, because when particle gets into a biological fluid, even when we inhale it, it first gets in contact with the lung lining fluid. So it's not really dry. Our alveolar surfaces in the lung are covered with fluids, which contains lipids, proteins, and so on. So foreign particle gets enveloped in those biomolecules. And this shell, this coat is called nanoparticle corona, a protein corona or biomolecular corona. And that corona actually modifies the way the particle will be taken into the cell, transported further, aggregate with the other particles or with the other biomolecules. So what we need to understand is the corona, and corona will be our main focus here. So we will try to understand how corona is formed, and based on this, make some predictions. And that is indeed possible. I will cite certain things, so certain papers that prove that such an approach can be successful. What are the questions we're addressing? First thing is most challenging, and it's the holy grail of entire physics. So basically to take atomic structure of a material and predict its properties at whatever scale. Certainly that's in the general sense, it's impossible. In some cases we can make it work, but it's a really grand question. But we ask now, what material properties determine their functionality, specific functionality, toxicity, or biocompatibility? Can we really point at those properties that can be predictive? How do the materials affect the processes in biotechnology, medicine, or food production? So how do we, should we coat, say, pipes in our dairy processing, milk processing plant to avoid fouling, for example, or avoid unnecessary transformation of proteins? How can we improve drug delivery with nanocarriers? If we understand the interactions at the surface, then we should be able to optimize the process. Can we avoid fouling? Can we make materials safe by design? So if we know what property is responsible for the adverse effects, can we actually reduce it during the production at the stage of design? So that's actually one of the ultimate goals that was set in these European projects. We want to help industry to save time, to avoid, say, the testing of materials for safety in vivo, and do simple and inexpensive in vitro and in silico tests before the material produced in large amounts. That would save time and money. So this is the kind of the overall idea. But the main question is how the physical and chemical properties of materials are related to their biological activity, to their functionality. So if we say we want to address it theoretically from first principles, then we face a lot of challenges. And some of these challenges we didn't recognize at the beginning, so we actually hit them during the project already. And then it took a lot more time then to overcome those, at least partially. We had to look for ways to overcome those challenges. And I'll show you some answers we got, not ultimate answers, but some working solutions we got to all of these challenges during this presentation. First of all, we know that there are only about 100 chemical elements, and about maybe 20 to 30 of those that are sufficient to build most of the material around us. So we don't really need to understand the huge diversity of that. And in principle, if we know the properties of all these atoms and how they can make materials, we should be able to make predictions. But if we talk about biology, certainly that's not sufficient. So even if there are only 10 different atoms in a protein, atom types in the protein, the diversity of proteins is immense. So diversity of biomolecules, diversity of interactions is immense. So we need to actually overcome this difficulty. And the responses are extremely complex. And quite often they're active responses, like ATP depending responses that we cannot really model at the physical level. Okay, so we need to find a way to address those complex questions, complex situations from first principles. Then we talk about immense system sizes. Theoretically, we can model an entire protein, but it's not really useful for the questions we ask. What we need is a nanoparticle with a corona, which can be the tens of nanometers, and relatively high binding energies, so which makes high barriers between different states, and system dynamics is very slow. So how do we overcome this? Then when we talk about this, we suddenly realize that there are two different worlds developing their tools in completely separately. The world of biomolecular modeling, where a lot of work has been done over decades, and a Nobel Prize already awarded, and world of inorganic materials, where a lot of work has been done, but you actually realize they talk different languages. You can't just take a common model for dissimulation of a protein and combine it with the common model of an inorganic material. They're actually using different force fields, which cannot be put together immediately. So lack of force fields is another challenge. Then lack of information about the structure of nanomaterial surface. If we're told that in an experiment like a drug delivery carrier is 5 nanometer size gold nanoparticle, we still don't know what the surface is, but at the scale of interaction with the protein, everything is important. What kind of crystal phase is exposed? Are there any defects on the surface? Is it rough or is it smooth? And it makes a lot of difference. So we need to make assumptions and get information from either from simulation or from other reasoning. Then water is a headache. You can imagine if you did molecular simulation or experiments, we cannot predict everything in water. We can tune the models to predict certain things. But if we are systematic, we can try to make predictions from first principles still and be consistent. And then finally, long-range forces. If you're doing molecular dynamic simulations, you know that most of simulations at the all-atom level are performed with the cutoff of 1 nanometer. But what if your particle is 10 nanometers size? How can you include the interaction with the rest of the nanoparticle? You can say, okay, that's not important, that's too small. No, if you do the calculation, you see that interaction with the rest of a 5 nanometer nanoparticle will be still say 10 kBt, which amounts to 20-30 kilojoules per mole, which is not negligible. So we have to deal with that somehow. Okay, how we do this? Some ideology. So this is how we address the challenge 1. I show you my say classification of challenges here and on the banners. It's a nanoinformatics approach. So if the system is extremely complex, behavior is extremely complex, and the parameter space is multidimensional, so we can vary various parameters, then statistical models, machine learning helps a lot. And at this point, we're not overly ambitious. We just say, okay, when it comes to real biological molecules, we do, not only biological molecules, but active responses, complex responses, we do statistical modeling. We measure those and try to do this by machine learning. But on the other side here, we need to collect the meaningful data that are predictive for that behavior. And one of the ways to collect data would be to perform molecular simulation. Then we put them in computed parameters into the database, and then use them as inputs to statistical models to make predictions. So this is the idea. Then overall strategy can be summarized as follows. I split the problem into two parts: physics-based materials modeling to evaluate the relevant properties as far as we can go. And then from that point on, data-driven modeling to predict functionalities or biological activities. So we're going to relate advanced descriptors calculated from first principles to complex functionalities. So here I have statistical and say from atomic structure to complex descriptors by an interface, I have physics-based. Then our scheme will be bottom-up. We want to be not dependent on experimental measurements, but do everything from first principles, even before the material is produced. Then on the biological side, our advantage is that we can use system structure and symmetries, like repeat units in biomolecules. We can recognize that there are only 20 different amino acids or sugar or lipid elements that can be modeled. So there are thousands or tens of thousands of proteins, but only 20 amino acids. And we want to pre-calculate the important interactions and use that. And then we'll feed the properties into statistical models as inputs. So I'm going to show you that it's indeed possible, what motivated us, and some of the publications that demonstrated that based on these advanced descriptors of nanomaterials, you can make complex predictions. So here in this paper, the group from Barcelona predicted cell uptake of different nanoparticles. There were iron oxide nanoparticles with different modifications. And they found that if you know certain advanced descriptors, they are not known immediately from all atom structures, but can be calculated relatively easily. Lipophilicity, van der Waals surface area are predictive of that. And if you know these properties, you calculate it systematically for a range of materials, then you can build a regression and make a prediction for the cell uptake. And it works. So you see the correlation is pretty good. So that sounds promising. And another one is a prediction of cell uptake based on protein corona statistics. So here what was done, corona of gold nanoparticles was measured, and then statistics of different protein properties included in the corona calculated. And you see, if you know that average amino acid percentage in those proteins adsorbed to nanoparticles is actually, or molecular weight, are predictive of cell uptake. And it's quite understandable because basic or say small amino acids determine actually how the protein interacts with the surface. It reflects the properties of the surface in some integral way that are responsible for that interaction. And this we see as a way to go. We don't start with the really basics, really atoms, but we start with some integral properties, basic interactions that reflect the type of the material and type of surface. Okay, so this is another promise like this. And we can imagine schematically our program as follows. We start with the certain proteins that we know appear in the system, either from corona analysis or from other things like cell receptors. calculate properties of proteins. Then on the other side, we calculate nanoparticle structure, nanoparticle descriptors. From combining those, we calculate abundance of that protein in the corona or binding strength, and then from that, predict nanoparticle activity. So that's the idea. Okay, so the overall algorithm, which is a really multi-scale and multi-stage, you see the outline here. So we start with the real nanomaterial crystal structure, which we generate using standard tools, then perform atomistic simulations to calculate certain properties, then feed it into a more large-scale model to do coarse-grain simulation. Then we feed it into interaction with the protein using a coarse-grain presentation of a protein to calculate the interaction. And then we study competitive adsorption. Then we study, concentration of proteins in the corona. And then finally, we come to the adverse outcome and certain biological activity. So you see, it's all like first principle pathway that we want to just to walk. Yeah. Good. So from implementation point of view, so main elements of our multi-scale model is atomistic simulation. You see a snapshot. We perform atomistic simulations of biomolecular fragments with surfaces, then simulations of entire proteins interaction with surface, then simulations of proteins competing for adsorption, and then we try to predict cell uptake or membrane penetration. And this approach is systematic so that we don't just take parameters from experiment or from elsewhere, but we feed in parameters calculated at the more detailed resolution to the larger scale system. So it's still mostly first principle part. Okay, now the scheme of coarse graining. So our main steps is coarse graining proteins. We do 1 bit per amino acid model, and then coarse grain it further. 2 levels of coarse graining for proteins. This is a united amino acid model. For nanoparticle, we just split it into 2 parts, core and surface, which we treat differently. And then using the 2 models, we can model adsorption. And then using this model and this model of nanoparticle, we can model competitive adsorption. So this one is used for single protein, and this one is used for multi-protein systems. How the systematic approach is built. Our starting point is all atom representation, all atom model. So we consider it as, well, it's not strictly speaking ab initio, but we can derive force field from that still. We can demonstrate that it's possible. But most of the time, we start with this kind of type generic force field, where overall system energy is presented as a sum of all possible pairwise interactions between the atoms. Then we group those and present this. We pre-calculate some of those, and some of others we just disregard. So we say bonding interactions, like bonds, will be intact during the processes we are modeling. So therefore, they contribute a constant energy. We're not interested in those. But what we model is a non-bonded part of energy, like van der Waals interaction and includes volume and electrostatic interaction. And for that, we use some approximations already. For electrostatic interactions, it's like the Debye-Huckel model. And then we say we group all atom energies into interaction of amino acid with a nanoparticle. So here, the overall interaction of a protein with a nanoparticle will be calculated as a sum of interactions of individual amino acids. And here, the parameters of that, we simply rotate the protein, explore all different distances and orientations. And certainly for each orientation, each position with respect to the surface, there will be a set of distances between amino acids and the surface. Okay, so we say we are not losing anything at this point, although we're already making some approximations. Further assumptions are, we assume that protein is a rigid body. We neglect its conformational changes. We can discuss it later, but it's a necessary approximation at this stage. Then we take conformation from optimal folding, ideally from crystal structure, but where it's not possible, from some generated structure. Then we assume pairwise additivity of all the potentials. And then we say energy of non-bonded interactions, electrostatic is here, but energy of other non-bonded interactions will be split in two parts. A surface, which will be modeled at the atomistic level, and bulk, which is modeled as the continuum level, core. My index here is core. Good, so I start now with the detail, how it's implemented. We first discuss how we model surface interactions. And for that, we need a lot of detail, because we want to take into account the exact structure of the surface, how atoms, partial charges are located, how it is hydrated. So hydration effects must be there. How charges interact with each other, how the bulky parts interact with each other, how the surface roughness even and the angstrom scale affects that. Good, and then if we say, if the force field is not there, for titanium dioxide, we first realized that force field that people use to simulate bulk properties of titanium dioxide are not really good to model the interface. So we thought we need to rederive it properly to take into account the surface hydration. So my challenge 3, how we address the force fields, is explained here. What I'm showing on the next few slides, it's a work from Sasha Lubartsev from Stockholm University, chemistry department, and they did really a lot on this. And this method is presented in our joint paper, which is archived, not yet published, but the manuscript is available there. So we start with the DFT, ab initio modeling of surface of a material like this in presence of water. So what is extracted from DFT modeling? First thing is different coordination, different atom types that arise from that. And you can see for titanium, there can be 3 different types, some hydrated ones with the water attached, some hydroxylized and some just uncoordinated with water sitting on the surface and the bulk atoms as well. So what is done? We collect statistics about the partial charges, electron distribution on different atoms, and then this gives us the information about how the force field and partial charges can be parameterized. The result is, from this ab initio simulation, is that what we need is 2 types of titanium atoms with different partial charges, 1 type of hydrogen atoms, and 5 different types of oxygens. It's like a bulk in titanium dioxide, then absorbed water oxygen protrusion, then in bulk water and oxygen bridge. So then the force field is parameterized as follows. For each of those atoms, we have partial charge and van der Waals parameters, Lennard-Jones interactions parameters, which will be compatible with the charm force field, or same force field used for simulation of biomolecules. Okay, so if we do this and extract this from quite an immense ab initio simulation, then we have a new force field that is parameterized from first principles and which correctly takes into account the surface properties, namely hydration of the surface. Okay, so when this is done, we can do make the next step and model the interface. This is my challenge 4. We then perform all atomistic simulation of an interface. Now, a block of material, a slab, is modeled in contact with the individual amino acids or segments of biomolecules in presence of water and ions, if necessary. Then what is calculated? We calculated the potential of mean force using metadynamics and Plumet methodology. Plumet software with the metadynamics methodology. What is done there? The interactions of the amino acid or the molecular segment with the surface compensated with the hills, Gaussian hills, until we get a flat really distribution of biomolecule with respect to the surface. And then some of those hills that compensate any potential wells or barriers in the real average potential, we invert them and get the potential of mean force out of that. So the result of this metadynamic simulation is an energy profile. What we do to represent the most important biological molecules, we model amino acid side chains or side chain analogs. Then we model also lipid fragments and sugars interacting with the surface. And for each of those, we perform this kind of metadynamic simulation to extract the potential energy profile, potential mean force. The result looks like this. So if you want more details, you can look into publications, but typically the system box is like this. So we have a slab of material, then water with ions, and then amino acid there. It's run for 500 to 800 nanoseconds overall. And we use GROMACS for simulation with the Plumed charm to form this system and crystal structures from a materials project to form this lab. What is calculated for different surfaces, what you see the results here is for silver, potential mean force for silver. And they indeed contain a lot of information. And then most of complexity is already integrated in. For example, on some of those, you see barriers, you see minima and maxima reflect the preferred hydration state. So some amino acids would prefer to have a water layer between themselves and the surface, and it is included in the model. So after that, we can run the implicit water model. Okay, then we say if we simulate it for a slab, for a flat surface, we need to correct it for spherical particles. And we do it using a simple continuous theory. This is how we handle our last challenge, the long-range forces. What if the surface is not flat? For flat, you see within the molecular dynamics cutoff distance, like 1.5 nanometers, we have only this kind of length part of the surface. What if we want to simulate it against cylinder or nanoscale sphere? Then the range of material, the chunk of material within the cutoff will be different. The shape is different and number of atoms is different. So we do simply geometrical correction. So we just calculate how much less material will be in the range and correct the potential mean force using this correction formula. Then after that, after we correct it, we can calculate already interaction of individual amino acids with particles of different size. Now we have to coarse-grain the proteins. Our coarse-grain model is as follows. So it preserves the shape of the protein, location of charges and dipoles as follows. We put a single bead for each amino acid and put it at the position of alpha carbon. And then we parameterize the interaction of each bead with that potential mean force that we calculated using atomistic simulations. And then construct the overall energy of interaction of the protein using those potentials. What if the three-dimensional structure of the protein is not known? And we underestimated this at first when we looked at the proteins observed in the corona, measured in the corona. We just realized for majority of them, like 80%, 3D structure was not known. So we went for this kind of homology modeling and structure prediction. So the best tool we found was an ITASR. We could use Rosetta as well for small molecules, but with the ITASR, we can run it and generate structures, three-dimensional structures here using just the sequences. So our starting point is a sequence from a FASTA sequence from UniProt database. Then we run the simulation. It takes about a day on a good computer to get the 3D model. And that one we record in the form of a PDB file and feed it into a coarse-grained model of a protein. Okay, for a nanoparticle, it's two-layer model. Surface is modeled using the potentials of mean force, atomistic simulation, but the bulk is modeled using Lifshitz theory. So we use Hammaker procedure. We simply integrate the interaction between each amino acid and the bulk atoms. So we don't calculate it pairwise for each pair of atoms, but we calculate it. Each amino acid, we know its volume, we know amount of material in there. So it's like radius of amino acid is entering. And integrate its interaction with the rest of the nanoparticle. If geometry is simple, like sphere or cylinder, we can pre-calculate these potentials. And this saves a lot of time, certainly. But this potential is long range. It's actually problematic to include it into real MD simulation. Okay, so when we do this, we still need to characterize a material. And we looked into the ways to calculate the Hammaker constants for different materials. So if we're taking titanium dioxide or gold or something, then we need to include the dispersion interaction based on the parameters of these materials. What we need to know is the refractive index for proteins. We know that from experiments. And say for refractive indices for materials, we can either calculate using DFT or know from experiments. So this is kind of the only experimental input for our simulations. But we need those to calculate interaction, the long-range part of interaction of each protein with the bulk of the nanoparticle. Okay, so I show you now the results. So I described the methodology. Interaction parameters. So Hammaker constants that we calculate based on those dielectric properties of material and amino acids look like this. For each amino acid, we have a number. Then we calculated them for a bunch of materials. This is for titanium dioxide, rutile, and this is for gold. And you see the numbers are, most of the time, are pretty similar. For some amino acids, they're higher, some are lower, but all in the same range. Then we perform this kind of calculation. So we rotate protein and move it up and down, close and farther from the surface to calculate overall interaction energy. And then we perform Boltzmann averaging. So we say out of all the studied configurations, in terms of distance and orientation, we calculate the energy and then calculate Boltzmann weighted average. It's like ensemble average. And this is what expected to be observed in experiment. Good. Then with that, using this methodology, we can calculate adsorption energies for individual amino acids and segments. And you see the result will look as follows. Then we get, as for example, for gold nanoparticle with different amino acids, we see that they are attractive. Arginine, for example, shows attractive energy of -18 kBt, which is quite a lot on the histidine and so on. So we can do this calculation. So these are already important descriptors of a nanomaterial for biological activity. So we consider this set of descriptors as a fingerprint. We can distinguish nanoparticles, nanomaterials from one another using these numbers and make predictions. Good. Then another outcome of the model is quite an impressive thing, is a heat map of energies. What's shown here on these heat maps is the energy of protein interaction with the nanoparticle. So it's serum albumin, human serum albumin on gold nanoparticles of two different radii, 5 nanometers and 100 nanometers. And what's shown on this heat map are the energies versus two orientation angles. We rotate protein like this, and then like this, around two different Euler angles, theta and phi. And you see each of them gives us a different energy, and we see the spots, like green spots, will be minima. And this is also quite an important information because it says what is the preferred orientation for the protein for binding on that surface. So we can make predictions, and this can be visualized. One of the outputs of the model is it spits out PDB of optimum configuration on the surface. And that's quite useful. So if you want to decorate the particle with the proteins, we'll understand how they bind. And you see that depends on the size. So this varies with size. Another thing that we can do, we can plot average energy, average of all orientations versus size. And this is for the first time available. This possibility appears only in the model that we've developed here. So we see that depending on nanoparticle radius, the adsorption energy changes, and this is another parameter of optimization. So if you want to design your carrier, this will be useful information. Okay, then the next thing, we can rank the proteins by binding affinity. And here you see a table from a paper from 2019, where we rank proteins, 10 different proteins by adsorption binding affinity to gold nanoparticles. And we see that indeed information can be useful. What we can predict in this way is what is expected to be in the corona, what will bind stronger, and how the content of the corona depends on the particle size. So this is a similar table, but we try to validate our model, whether ITASR prediction of three-dimensional protein structure is good enough to make predictions of adsorption energy. And we compare energies for proteins folded by ITASR versus PDB, the crystal structure, crystal protein with crystal structures used for this calculation. And prediction is reasonable. It's not exact. There are deviations, but we found that it's good enough. So ITASR is doing a good job, at least for binding affinity. So what we try to compare is using the experimental crystal structure and ITASR structure computed protein three-dimensional structure to compare ranking. It's not exact, but it's quite stable, quite robust prediction. For example, for gold, we see also the order is slightly different, but 6 out of 10 proteins are the same in the top 10 binders. For titanium dioxide, it's 5. And we can calculate numerical measure of that prediction. And we see that Kendall-Tau correlation coefficient compares to ranking tables on how well they match each other. And we see 66%, 60% or so is predicted correctly. So most of that is working well. But you can see it visualized on these graphs. And we plot adsorption energies predicted from PDB structures versus ITASR predicted structures. And you see the correlation, although it's not perfect, but quite reasonable. And you see the deviations are mostly within like 10%. Some proteins, though, they deviate by 20 or 40% from between the two models. And this is from paper published just this year. Good. And then we try to use this statistical model. We try to use meta models to make predictions. We see overall calculation is pretty heavy, but the question is, can we actually predict those adsorption energies using just protein characteristics and nanoparticle characteristics? And the answer is yes, we can. This is also from that paper published in the Nanomaterials. And what you see there is a result of neural network, where we train the neural network to predict the value of adsorption energies for protein just from the protein statistics. So say for the types of amino acids, the counts of amino acids, and the folded structure of a protein. For example, how many of them are sitting on the surface. And it's indeed possible. On the training set, it works well. So most of them can predict within 10%. And on validation set, which is 20% of that, we see we're still pretty good. So we can really predict it with a reasonable accuracy without doing all this calculation. But we need certainly a lot of data to train the model well. Okay, so this is possible. But then what about our final goal? Yeah, so on this slide, I wanted just to mention that this protein corona model that we've developed, starting from potentials of immune force for specific materials and for proteins, is free to use. So if you register on nanocommons.eu website, it's website of the project, NanoCommons knowledge base. One of the tools integrated in the knowledge base is a Corona tool. You can pick a nanoparticle from the list of materials, and you can supply a name of a protein in UniProt database, and it will calculate you the adsorption energy and heat map. Okay, so then we can study adsorption as well, in competitive adsorption, when we have those individual interaction energies. And our way to go is to coarse grain it even further. Even the 1 bit per amino acid model is too heavy. So what we do, we group them, about 50 amino acids in each block, in each bead, and then parameterize interaction of these beads with the surface. And the mapping criterion is as follows. So we take the heat map calculated in this higher resolution, 1 bit per amino acid, and optimize the potentials for these large beads to match the heat map. You see that it has proper shape, proper energy in that, and proper preferred orientations. So all these things are preserved. And when we parameterize it in this way, we can do the next modeling and study really competitive adsorption. We see that it worked. We see a Roman effect, for example, replacement of small proteins by large ones on the surface, and so on. So we have now these models. We haven't used them a lot, but say we can extract, for example, adsorption profiles result in time. And final thing is, if you want to apply this for predictions of biological activities, what we do, we collect those advanced properties, like, for example, binding energies for molecular groups, say hydration energies, rank on a binding scale, and so on, and make predictions. Two examples I show you from Smart Nanotox project here. One is we take in vivo inflammation data that were measured on mice exposed to nanomaterials. So there are about 40 nanomaterials in here, 40 in vivo experiments. And they measured the degree of inflammation, count of neutrophil cells that come in when inflammation starts in the lung. And what you see on this graph is a set of points measured on the vertical axis is measured and predicted on the horizontal axis. And prediction is made using neural network trained with the input data from our computed characteristics of bionano interface, interaction energies. Another more advanced model, this was a random forest model using the QSAR workbench from BioVia, Dassault. Marc Minier, who worked with the Smart Nanotox project, did this modeling, and you see quite a good prediction again. So this is the same inflammation prediction of inflammation in vivo using various descriptors of nanomaterial in interactions. So the correlation is pretty good. So you see here, but on the right, what you see is a kind of parameters that were found important for this prediction. And in addition to just biological settings, we have like surface area of material and various adsorption energies. We see, for example, that adsorption of a lipid is quite predictive of the toxic outcome of this interaction. And one of the recent publications, this paper is now already published online in Advanced Materials. That is indeed, I'm really proud of this work. It's a result of Smart Nanotox project. 8 labs collaborated in this to do this. And it is a completely mechanistic description of interaction of nanoparticles with the lung, predicting chronic adverse outcome and nanomaterial cycling, various reactions at the cellular and molecular level that are really predictive, and that understanding mechanistic picture of this development of an adverse outcome allows us to build models, simple in vitro and in silico tests that can tell us whether the material will produce this chronic lung inflammation or not. So we can make this prediction of long-term effects just based on really simple in vitro and in silico tests. So we actually demonstrated that this kind of overall big goal is achievable. So finally, I summarize and give an outlook. So we see that if we want to make predictions of complex outcomes and functionalities of materials, we should combine physics-based modeling and data-driven modeling. So we cannot do everything from physics-based, and we cannot do everything using just the data-driven, but the combination worked well. Then for physics-based, to describe complex interactions properties of bionano interface, we need to use a multiscale methodology, combine different resolutions and different levels of description. Then we see that bionano interface data indeed have predictive power, so we can predict real biological events using that. And then we can also validate it using measurements at different scales. So we try to validate it using either protein corona measurements or biological activities, cell uptake, and other activities. So in principle, there are many questions, certainly, but we can answer and we can justify the path we've chosen in this way. So that's it. Thank you for your attention. I'll be glad to answer any questions if